In this video, we will cover what will be on Quiz 2. So the following activities will be found for Quiz 2. Quiz 2 will cover the standard plate count. It will cover the endospore stain, and you should know an example of an endospore positive bacteria. And the example that we give you is going to be Bacillus anthracis, which causes anthrax. You should know the acid-fast stain and an example bacterium for that. That would be Mycobacterium tuberculosis, which causes tuberculosis. This will also include the OF glucose test, so our oxidation fermentation test, our Fennel Red Test, and our IMVIC. And so what I'll do is I will go through the PowerPoints for each of these activities and tell you kind of what are the things that you should focus on for Quiz 2. So let's start with our standard plate count. So for your standard plate count, you should know the purpose. And the purpose of the experiment is to isolate colonies of bacteria from a large population. So isolation is important. And determine the titer of the stock culture. And then you should know what a titer is. And the titer is the number of cells per milliliter. And so the other thing that you want to know from this is that we can't use a streak plate to quantitate the amount of bacteria. A streak plate can be used for isolation, but it's not used to determine our titer. So one of the things that you want to know for your standard plate count is how to calculate both your dilution factors, meaning the amount you dilute each time. You should also be able to calculate the final dilution factor, both for the tubes in the serial dilution as well as on the plates. So you should be able to do that math. You will not be given a calculator. It's all in multiples of 10. So that math should be done with no problems without a calculator. You should be able to read a micropipette if given a volume shown, and then tell me what volume the pipette is set to. And so again, if we look at this, a P1000, for example, the top placeholder would be the thousands, then the hundreds, tens, and so on. For a P200, the top number is going to represent the hundreds, so 200 is set to a volume of 200. A P20, the top number is going to be the tens placeholder, then the ones, and then after for the decimal. And so you should be able to look at these micropipetters and tell me what volume the pipette is set to if I tell you which pipette it is. Like I would have to tell you that this would be for a P200. So I will tell you what pipette it is, and then you would need to look at the dial and figure out what volume it's set to. You should also know the minimum and the maximum for each of the pipettes. So again, if it's called a P1000, the maximum is going to be 1,000 microliters. The minimum is going to be 10% of that. 10% of 1,000 is going to be 100. So for each of those pipettes, you should also be able to tell me what is the minimum volume you can use and what is the maximum volume. So again, this is just showing you how to read a micropipette. Same thing here, just more practice reading a micropipette. You should understand the general parts of a micropipette, right? That this is going to be the push button and that you should only push to the first stop when you go to draw up the liquid. You only push to the second stop when you're trying to get the liquid out of the pipette. You should know that this is the thumb wheel. This is what you can turn to change the volume. We have our tip eject button, and then the barrel of the pipette is going to contain where you would put the tip. And so these are just some general things that you should understand about a micropipette. You should understand the general idea of how to use a micropipette. So you need to twist the dial to the desired volume, add a tip, push the plunger down to the first position, then put it into the liquid. Slowly release your thumb to draw the liquid into the pipette. Move your pipette tip into the desired location, so if it's a tube or whatnot. And then when you go to get the liquid out, push to the first stop, then the second. Take the tip out of the liquid, and then release the thumb. and then you can eject the tip. So you should know kind of the general steps when using a micropipette. There's nothing that you need to know for this part. You don't need to memorize the materials, what is needed for this experiment. You should know kind of an overview of some of the basics of the standard plate count. For example, you don't want to take these nutrient agar deeps out of the water bath all at once, because if you do, they will polymerize, meaning they'll solidify before they get poured on a plate. So you should understand that you should only take one at a time. You should understand that your plates should be poured close to the flame. That's your sterile field. And that once you pour them, that you don't move them. Other than that, there's not much else on the slide that you need to know. You should understand just kind of general rules for how to do a standard plate count. So what are the things that you should do to maintain your aseptic technique? So just review these to have an idea of what to do. For day 2 of our standard plate count, again, this is just reviewing your serial dilutions. We did an example together in the Zoom discussion of an additional standard plate count serial dilution. So go back and review your notes for the dilutions that we calculated. We calculated the dilution factors. We calculated the final dilution factor of each tube and each plate. And so go back and review your notes for that and make sure that you're familiar with how to do those calculations yourself. So on this slide, you want to know what CFU stands for. CFU stands for the colony forming units, which is basically the number of colonies that develop on the plate. You want to know that you need to count the plate that has between 30 and 300 colonies. You don't want to count less than 30. That's too few to count. You don't want to count more than 300. That would be too many to count. And so you should know that you should pick the plate that has between 30 and 300. And so in this example, you can see that this plate here, this 10 to the negative 6 plate, has 82 colonies. That would be the plate you would use to do your calculation, because that's the plate that's between 30 and 300. You should understand that how do you calculate the original cell density, right? The original cell density is going to be your colony forming units, so the number of colonies on your plate, divided by your final dilution factor, the one that's actually written on the plate. And so if this was my calculation here, 82 colony forming units divided by my final dilution factor, which would be 10 to the negative 6, and then again, you're going to multiply by the positive exponent. So 82 divided by 10 to the negative 6 is the same as 82 times 10 to the positive 6. And then you have to put it in proper scientific notation. So you would move your decimal point one place, and then you need to change your exponent one place. And so you would end up with 8.2 times 10 to the 7th CFUs per milliliter. You want to understand that your units would be in CFUs per milliliter. So you would, if given a set of pictures of plates that would be found in a standard plate count, you should be able to, if given the number of colonies like is shown here, you should be able to calculate the original cell density. And again, this is something that we reviewed during the Zoom session. The other thing that you want to understand is that each one of these colonies that forms on the plate, each one of those colonies represents one initial bacteria. And that one initial bacteria reproduced asexually. It made copies of itself until it got to millions of bacteria that you can see as a colony. And so that's why each colony is what we call a colony forming unit, and that represents one original bacteria that was plated on the plate. You also want to have a general understanding, and this was something I talked about in the video, about why the colonies are different sizes. Why are some of the colonies so much larger than other colonies? And that was because I told you that the bacteria that's growing aerobically, meaning on the top of the agar where oxygen is available, that bacteria is going to grow faster than bacteria that's growing within the agar, because remember, you mix the bacteria with the agar and then pour it. So bacteria that are in the middle of the agar, they're not getting access to oxygen, and therefore they grow slower. And they grow slower because when they're grown anaerobically in the absence of oxygen, that means that they will only make, for fermentation, 2 ATP per glucose. When they grow aerobically, so in the presence of oxygen, they will get between 30 and 32 ATP per glucose. So the ATP yield is a lot higher when bacteria are grown aerobically, and that as a result, they will grow faster. So now we'll move on to the endospore stain. And so for the endospore stain, you want to know the purpose, and the purpose is to stain and visualize endospores and differentiate spore-forming cells from non-spore-forming cells, and this is a differential stain. So you want to know the purpose. You want to understand when endospores form. And endospores form when conditions become harsh. And so you want to know these different conditions for when bacteria would experience their condition as being harsh. So when temperature changes, or pH, or dehydration, or UV, or nutrient loss, or changes in oxygen concentrations, that is a condition that bacteria would experience as being harsh. And when they basically experience these harsh conditions, they will go through the process of sporulation, meaning that they will form an endospore. You want to know that the endospore is made of the protein keratin, which is found in skin. And so because of that tough keratin protein, it makes the endospore very resistant to these differences in changes. They're very heat resistant, resistant to chemicals, etc. So you want to know that endospores, what they're made of, what types of conditions would cause bacteria to form endospores. You want to know that eventually the vegetative mother cell will break down. It will give rise to a spore, and then when conditions become favorable, the bacteria will undergo germination and will go back to being a vegetative cell. And this cell is the one that's metabolically active. It's the one that's actively dividing. So you want to know the purpose of the endospore. What is the name of the process when they form the endospore? What is the name of the process when they go back to being a vegetative cell? What is the endospore made of? What are the harsh conditions, etc. In terms of examples of endospore producing bacteria, again, you don't need to know the whole list. I gave you the organism that you would need to know, and that is that Bacillus anthracis would be your example organism of a bacteria that produces an endospore, and that causes anthrax. You also want to know that there are 2 genus of bacteria that are most commonly going to have endospores, and that is Bacillus, which is aerobic. So you want to know this, Bacillus that's aerobic, and Clostridium, which is anaerobic, and that both of these genus are gram-positive bacteria. So more commonly, endospore-producing bacteria will be gram-positive. There are some exceptions, but generally speaking, endospore-producing bacteria are the ones that are gram-positive, and usually Bacillus or Clostridium. Now, in terms of the staining procedure of the endospore, you don't need to memorize the steps. However, there are some keys that you need to know, and that is that you have to apply steam when the malachite green is added. And that's because you need that steam to get the dye into that tough keratin protein. And so we steam our slide to get the dye to go into the endospore. So that you should know. You should know this diagram. Basically, the general understanding of when we apply the malachite green, that both endospores and vegetative cells will both stain green. When we decolorize, you don't have to remember that you decolorize with water, but you should know that if you decolorize, that the endospore will stay green, the vegetative cell will become colorless, meaning that the dye will come out. And then you should know that we use safranin as a counterstain. Again, you don't have to memorize the time or anything like that, just that you use safranin as a basic stain, and we don't apply heat, and the safranin is going to stain the vegetative cells, so that when you're finished, you end up with red, pink vegetative cells and green endospores. So again, you don't need to know the specifics of the procedure, but you want to understand an overview and how that affects the colors at the end of this experiment. You should be able to recognize a picture as being an endospore stain. You don't have to memorize that this was Bacillus polymixa. You're going to use the example organism of Bacillus anthracis. That's the one you want to study. So you don't need to know which organism produced the slide. You should just know that this would be an example of an endospore stain because we have that teal green endospore and we have our red pink vegetative cells. And so this would be what an endospore stain would look like. Now, moving on to our acid-fast stain. You want to know the purpose, right? And the purpose is to stain acid-fast bacterial cell walls and differentiate acid-fast positive bacteria from non-acid-fast bacteria. Again, this is a differential stain. You should know that organisms within Mycobacterium would be acid-fast positive and that they have a cell wall that is made of 60% mycolic acid. And then you want to study what are the purpose of the 60% mycolic acid. What advantage does it give bacteria by having this mycolic acid in their cell wall? And one, it makes the bacteria sticky, so it's an adherence factor. It allows bacteria to adhere to surfaces. It's sticky and it's waxy, which protects from phagocytic digestion, so meaning that the immune system doesn't break down that bacteria, protects the bacteria from dehydration. So again, bacteria can survive longer on surfaces as a result of having this mycolic acid. It makes bacteria resistant to chemicals, both antibiotics and disinfectants. So you want to know what is the acid-fast cell wall made of and what is the purpose of that acid-fast cell wall. I won't ask you to differentiate between the Ziehl-Neelsen method and the Quignon method. This was just more kind of for your information, and so you don't need to study the differences between these. In terms of the procedure, again, you do not need to memorize the procedure for this. You should, however, understand that we have to steam with carbofuscin. And the reason that we have to steam with carbofuscin is we need that carbofuscin to be able to penetrate that waxy mycolic acid. And so if we don't steam with carbofuscin, acid-fast positive bacteria will not stain because you need that steam to get the dye in. So you should know that we use carbofuscin with steam. You don't need to know which decolorizer and for how long for the acid-fast stain. You should know we decolorize. And then you want to know that there's a counter stain and that the counter stain is the methylene blue. Again, you don't have to memorize the procedure, but just know that we add methylene blue so that we can stain non-acid fast bacteria. And the next slide will show you why. So again, this is just an overview of the staining procedure. You don't need to memorize this diagram. You should, however, be able to tell me what acid-fast positive bacteria and acid-fast negative bacteria would look like if given these different steps, right? So that initially when we add the carbofuscin, both cell types are going to stain pink because the carbofuscin is going to get into the cell. Then we decolorize, and when we decolorize, acid-fast positive will retain the carbofuscin, Acid-fast negative will lose their stain and they will become colorless at this point, which is why we need our methylene blue, because the methylene blue is our counter stain and it's used to stain the acid-fast negative. We do not apply heat when we add our methylene blue. And so the methylene blue will only get into the acid-fast negative and not the acid-fast positive. And so you should know at the end of this procedure, acid-fast positive bacteria will be pink. They're kind of a magenta, like bright pink color, where Staph epidermidis would be blue, or the acid-fast negative. You don't have to memorize that example, but that the acid-fast negative would be blue because it would lose its carbofusion in the decolorizer step. So again, in terms of what organisms you need to know for this, you don't need to know Mycobacterium leprae or Mycobacterium smegmatis, but you should know an example organism for an acid-fast stain would be Mycobacterium tuberculosis, which causes lung TB. You should be able to recognize an acid-fast stain. So you would want to recognize that this is an acid-fast stain. Notice we have our blue cells and then our magenta cells. The blue cells are the acid-fast negative. The pinkish color cells are going to be the acid-fast positive cells. Now, sometimes students panic because they think that this picture looks similar to the Gram stain, and you might be right. So oftentimes, if I give you this picture, I might give you some other detail that would help you determine that it is an acid-fast stain and not maybe not a Gram stain. For example, I could say something about, you obtain a sample from a sputum sample, which remember is that coughing to get the fluid from the lungs. And that sputum sample might indicate to you that it's likely going to be an acid-fast stain. So there would be some key that might help you differentiate between the acid-fast stain versus the Gram stain, because they do look somewhat similar. Not quite, because these are blue and the Gram stain, they're purple. And then the pink is very different. In the acid-fast stain, it's kind of magenta. It's a bright pink, which looks different than in our Gram stain where it's like a light pink. So just be aware, you should still be able to recognize this would be an acid-fast stain picture. So now we're moving on to Lab Module 2. And Lab Module 2 is dealing with our biochemical tests. So one of the things that you're really going to see throughout this part is that you are going to need to study those biochemical sheets. And those biochemical sheets are something we're going to review, but basically you need to know the substrates, the enzymes, the products, if a pH indicator is used, if a reagent is used, what does a positive test look like, what does a negative test look like, etc. So for the Fennel Red Test, you should know that the purpose of this test is to determine if bacteria can metabolize a specific carbohydrate. And that carbohydrate can change. We can use pretty much any carbohydrate. We could have lactose in the media, we could have glucose in the media, any particular carbohydrate. You don't have to know the 4 bacteria that we used in this experiment. That is not important. So for this slide, in terms of what you need to know for this, you don't really need to know anything for this, except that the purpose of the Durham tube, that tube that's inside one of the tubes, that glass tube inside there, is used to detect the production of gas. Because some bacteria, when they utilize sugar, they will produce gas. And the Durham tube will allow us to visualize if gas is produced. You don't have to memorize that the plastic cap was the glucose and the metal cap was the lactose. That's not important either. So for day 2 of our Fennel Red broth, now when you're studying, students kind of panic about this in terms of, do I need to memorize all of the ingredients in every recipe? And the answer is no. So you do want to understand that in the Fennel Red broth test, that there are peptones and that in that media, the peptones are there as food for general growth. Basically, it's providing these chemicals, these nutrients, like a source of nitrogen, for example, that bacteria would need to grow. And as a result, in a Fennel Red broth test, you should see growth to say whether it's a positive or a negative. If you get no growth, we say that that is inconclusive. And I'll come back to that in a little bit. You should have a general understanding that sodium chloride is there for salt balance because, again, you don't need to memorize it for this recipe, but in general, that the reason sodium chloride would be in media is for salt balance because, again, you're going to see that over and over again. You should know that the media contains a carbohydrate because that is our substrate in our experiment, meaning that's what we're testing if bacteria can metabolize. And our carbohydrate is only going to have one specific sugar per tube. So in our experiment, we have one tube that has glucose, and we have another tube that has lactose. You want to understand that in this test, we use Fennel Red, and Fennel Red would be put down as your pH indicator. And then you want to know what the different colors tell you so that you know that yellow is acidic. Remember that for everything except methyl red, every other pH indicator we use, yellow is acidic. Red, right? So like a pinkish red magenta color would be on the alkaline side. So this would be our uninoculated tube. It's kind of this, this is an orangey red. So that's why it says red orange. That's the neutral. Whereas the pink, so kind of a magenta color, is going to be the alkaline. So this is used to help you fill out your biochemical sheet. So again, you want to study your substrate, your enzyme. In this case, it's various endoenzymes. We're not testing any one particular enzyme. You want to understand what the products are. So the products of this process are acids, alcohol, plus or minus gases. So those are your products that you're going to record. You want to record what does the positive test look like, right? So the positive test would be yellow with turbidity. You want to record and understand what does the negative look like, and that is that red pink with turbidity. So the bacteria grew, but they did not use the sugar. Instead, they produced alkaline products, which tells us that the bacteria used peptones as food. You need to know that no growth, if you see no growth in this test, no growth, the interpretation would be no result or inconclusive. We can't say if the bacteria can metabolize the sugar because it didn't grow. And we can make that assumption because this is complex or undefined media where there are multiple food sources. Bacteria should have been able to use something to grow. And so this slide is really important in terms of your understanding. You want to understand why your readout needs to be done between 24 and 48 hours. So that's an important part. And so the reason that your readout has to be done between 24 and 48 hours is because when bacteria are given the choice between metabolizing proteins or sugars, if they can metabolize the sugar is going to be their preferred food source. That's what they're going to use first for food. As they do that, they will start to lower the pH, and the pH will go down, and that's why our yellow is going to be acidic, and that's going to be our positive. But once we get to about 48 hours, the sugar is going to begin to run out because as the bacteria are growing, they're utilizing that sugar as a food source. That sugar is going to run out, and then bacteria have no choice but to then use the peptones as a food source. And remember that when bacteria use peptones as a food source, it's going to produce alkaline products, and the pH is going to go back up, and the tube is going to go back towards being orange, or if long enough, it could turn magenta. And so if you read this out at, let's say, 96 hours, you would see a tube that's pink, and you might say, well, that's a negative for the metabolism of the sugar. Well, no, not really. It's just you waited too long, And the bacteria then metabolize the proteins, which is what caused what we call a pH reversion, meaning the pH went back the other direction as the bacteria started to metabolize that protein when the sugar ran out. So you want to understand the importance of why you need to do this readout between 24 and 48 hours. You do not need to memorize the results of this experiment. I will not ask you, what was E. coli's reaction or what was Proteus vulgaris's reaction. That I don't care. What I do want you to take away from this, though, is if you were shown a tube like this, you should be able to tell me what that tells you, meaning what is your interpretation. So if we see yellow with turbidity, right? Yellow with turbidity is going to indicate that the bacteria grew, right? The yellow tells us that it is acidic. And if we look in the Durham tube, we can see the gas in the Durham tube. So acidic and gas is going to be positive for the metabolism of glucose because it produced the acidic products, which then tells us that it metabolized the sugar. If you see tubes that are pink, so we see this turbidity and it's pink, that tells you that this tube is going to be alkaline. That's the K. It's alkaline, which is a negative for sugar metabolism. Because if we see alkaline products, that means that the sugar was not metabolized, but instead the peptones or the proteins were used. And that produced the alkaline products, which raised the pH. You want to understand what it means to say that bacteria are non-saccharolytic. And non-saccharolytic means that they don't use sugar as a food source. So Alkaligenes faecalis would be a bacterium that is non-saccharolytic. It doesn't use sugar. Instead, it's going to use protein as its food source. So again, you do not need to memorize this image. You do, however, want to be able to interpret. If you were shown this, you should be able to tell me what your interpretation would be. So what is the pH like? What does that tell you that the bacteria use for food, etc.? So this is basically your biochemical sheet. And so for each and every biochemical test that we do, so Fennel Red broth, our OF test, our indole test, our methyl red, Voges Proskauer citrate, etc., you want to make these biochemical sheets for that. And so for each of those tests, you should know the name of the test, the name of the medium, the substrate, the enzyme, the products. What does a positive look like and why? What does a negative look like and why? Is there a reagent in that experiment? Is there a pH indicator in that experiment? And if so, what is it and what does it detect? And then additional notes. This would be where you would put other things that were talked about in the slides. So for example, in the Fennel Red broth, under additional notes, I would include the purpose, right? What's the purpose of that test? What does it mean to say that bacteria are non-saccharolytic? Basically, you want to understand your interpretation or your readout. Why do you need to do the readout between 24 to 48 hours? I would put that in the additional notes as well. So now we're going to move on to our oxidation fermentation or OF test. So for this test, you want to know the purpose. And the purpose of this test is to determine whether bacteria can oxidize and or ferment glucose. So not only are we looking at do they metabolize glucose, but do they do it in the presence of oxygen, which is oxidize, or do they do it in the absence of oxygen, which would be our fermentation? So for this, you want to understand that these are agar deeps. So they are a semi-solid. And so when I inoculated this, I had to stab inoculate with a needle. You want to understand what was the purpose of putting mineral oil on one of the tubes. And the purpose of that is that the mineral oil is used to seal out oxygen. This is your anaerobic tube, meaning your tube without oxygen. And this one is testing for fermentation, while the tube without the mineral oil is testing for oxidation. And so that's what you want to take away from this slide. So when we look at the readout, again, we have our recipe here. You don't have to memorize every ingredient that's in there. Again, you should understand a general idea that the purpose of a buffer is to keep the pH constant. You want to know that the agar is there as a thickening agent. Again, it has a low concentration. It's a semi-solid. And the purpose of that is that the agar is going to help to exclude the oxygen for the fermentation tube. You want to know what pH indicator was used in this test. So our pH indicator in this test is your bromothymol blue. Again, the blue tells you the alkaline color. So blue is alkaline. Yellow is acidic. Again, for every pH indicator we use except methyl red, yellow is acidic. So yellow is acidic. And then halfway between blue and yellow is going to be green. Green is neutral. And you want to know that our test has glucose because glucose is our substrate for oxidation or fermentation. So for this test, you want to know, again, your biochemical sheet. And so your substrate is going to be your glucose. Your enzyme are going to be various endoenzymes. So again, it's not just testing one enzyme. You want to know that the aerobic tube, the one that has no mineral oil, the one that's testing oxidation, meaning oxygen is present, that product is going to be acids. However, when we are looking at the anaerobic tube, the one that's sealed with mineral oil, that is testing fermentation. And when fermentation occurs, the products would be acids, alcohol, and plus or minus gases. So you want to understand the difference between the oxidation and the fermentation. You do not need to memorize which organism gave you which result. Again, the importance though is that you understand the readout. So if given tubes, you should be able to tell me what that tells you. So for example, in this one, yellow on the top, green on the bottom, and green throughout on the one with mineral oil tells me that it is positive for oxidation, negative for fermentation. And so you should know this term, which is that if you see an organism like this, that it would be a strict oxidizer of glucose. If you see yellow, yellow throughout, so both tubes are going to be yellow, yellow. Again, you don't need to know which organism, but you do need to know that that means it's positive for oxidation. It is positive for fermentation. So this organism would be an oxidizer and a fermenter of glucose. And then you want to know that that's referred to as a facultative anaerobe. And a facultative anaerobe will grow with or without oxygen, but grow better with it. And then lastly, we have our last set of results where we see blue on the top here, green on the bottom. This one is green throughout. So for oxidation, it's negative for oxidation. It's negative for fermentation because neither tube is yellow. So that organism would be a non-oxidizer and non-fermenter of glucose. And again, that term would be non-saccharolytic. They do not use the sugar, but they did use the proteins aerobically because the top is blue, meaning the pH is alkaline. And so that alkaline pH means that the organism used the peptones. And so you want to understand the readout, but again, you don't need to memorize which bacteria gave which result. I won't ask you that question. So this is just showing you bigger images of those results so that you could see them a little more clearly. So again, you can see that the E. coli is yellow and yellow. You can see that the Pseudomonas aeruginosa is yellow on the top. So again, positive for oxidation, but negative for fermentation. Alkalogenes faecalis is negative for both, but we do see the blue on the top, which means that they use the peptones aerobically. And so the last part of this is going to be looking at our MVIC. And so for your MVIC, you don't need to memorize which bacteria were used in this experiment, but you should know that there are 3 tubes that need to be inoculated. We have our MRVP broth, we have what's called our tryptone broth, which you'll see in another slide, and we have our citrate slant. You want to know for the citrate slant that you use a needle to inoculate it. You do zigzag inoculate the slant, but you need to use a needle for a citrate slant because you don't want to transfer too much bacteria. That will affect your interpretation. So now let's look at the readout. So you should know that the MVIC is the second most important ID procedure after the gram stain. So again, this plays a very important role in identifying which bacteria is present. You should know what each of the letters in MVIC stands for. So that the I stands for the indole, the M stands for the methyl red, the V stands for Voges Proskauer, and the C stands for citrate. So you should know what those different letters stand for. Again, you should know the name of the media. Those will be used on your biochemical sheet. So for both the MR and the VP biochemical sheet, the name of the media would be the MRVP broth. For the indole test, the name of the media would be recorded as being a tryptone broth. That's the name of the media. And for the citrate slant, the name of the media is a citrate slant, or more specifically, you'll see it's called a Simmons citrate agar slant, but citrate slant would work as well. So for this experiment, you would want to understand the readouts. This slide is a summary, but I will go into this in more detail for each one of these tests. So you don't have to worry about studying necessarily this slide specifically. So for your indole test, you want to know the purpose, right? The purpose is to determine whether bacteria can produce indole from tryptophan, and tryptophan is an amino acid. So you want to fill out your biochemical sheet, and that is that your substrate is going to be your tryptophan, and tryptophan again is an amino acid. This is my substrate. The enzyme that would need to be recorded is tryptophanase, which is an endoenzyme. It's an enzyme that's going to stay within the cell. For the products, you're going to record that there are 3 products. We have indole, we have ammonia, and we have our pyruvate. You want to know that this test cannot use a pH indicator. You can't use a pH indicator because the pyruvate is an acid and the ammonia is a base, and they cancel each other out, and you won't be able to detect a change in the pH. So for this test under pH indicator, you're going to put none and then describe why. For reagent, you're going to record the reagent is going to be your Kovacs, and you want to record that it detects the presence of indole. So that's what this reagent will react with. Kovacs alone is a pale yellow color. So the negative for this test is that the tube stays yellow, which means that no indole was produced, and bacteria do not produce tryptophanase because no indole was produced. The positive is going to be that pink red, and if we see a pink red, it tells us that indole is present. So the Kovacs reacted with the indole, and it turned that pink red, which is our positive. So this is basically your, again, biochemical sheet. You don't need to memorize which organism did which. So you don't need to memorize that E. coli was the positive and Enterobacter is the negative. But you want to know if you were given this result and you were told that this was an indole test, or you were told that this was a tryptone broth, then you would know it's the indole test, and you would know that this one with the pink red is the positive, and the one with the yellow is going to be the negative. So for your methyl red and your Voges Proskauer, so you have 2 separate biochemical sheets for this. They do have the same purpose. So the purpose is to determine whether bacteria can ferment glucose into mixed acids or acetoin. So we're looking at what type of metabolism do they do. In this test, our glucose is our substrate. So our enzyme is going to be various endoenzymes. And then for the methyl red sheet, the products are going to be our stable mixed acids. These are your products, and those stable mixed acids will decrease the pH. On the biochemical sheet for the methyl red, you're going to record the pH indicator and the reagent as being methyl red. In this case, methyl red acts as both because it's added after we do the incubation step, and it also changes color depending on the pH. So you would want to record that the pH indicator and the reagent is methyl red, and that methyl red is going to change color if the pH decreases. So it will turn pink red if the pH decreases. For the VP test, you want to record that at 24 hours, acids would be produced. So this is why we can't do our readout for VP at 24 hours. We have to wait the 48 hours. And so at the 48-hour time point, the bacteria will take those acids and they will convert them into a product called acetoin. And that product is what is going to be detected in the VP test. And acetoin is going to be a base. It will increase the pH. And so we add the VP reagents. It's called VPA and VPB. And those reagents will change color, meaning they will turn pink red if acetoin is present. So they will detect the presence of the acetoin. They're not a pH indicator. They are a reagent. It's the VPA reagent and VPB reagent. Again, for the VP test, there is no pH indicator. So for this slide, again, you want to understand that you cannot do your readout at 24 hours because for VP positive organisms, they will produce acids initially. So you want to understand that for MR positive organisms, that they will produce stable mixed acids, which will keep the pH low. For the VP positive organisms, that they will produce acids initially, which lowers the pH, but then will convert to acetoin, which will then raise the pH, and then the VP reagents can detect the presence of the acetoin. And so you want to understand question number 90 that was in the question set. So why are organisms that are MR positive usually VP negative and VP positive usually MR negative? And so I explained in that if an organism is VP positive, then it means that they produce acetoin, right? Because VP reagents will detect acetoin. So if they are VP positive, then they produce acetoin. Acetoin would raise the pH of the solution and therefore would not test positive with methyl red because methyl red only changes color if the pH goes below 4.4. So that's why organisms are usually one or the other, but not both. So for this particular slide, what you want to understand is that you inoculated the MRVP broth and then you allowed it to grow for 48 hours. And then you had to transfer the broth into 2 tubes. One tube got the methyl red reagent, one tube got the VP reagents. You do not need to memorize all of these acids and all of these products that can be produced for MR positive. You just want to know that for MR positive, they produce stable mixed acids. So MR positive, right, your positive result in this case would be if it's pink red. And if it's pink red, that tells us that acids were produced and the pH was less than 4.4. So that would be MR positive. MR negative would be not pink red or yellow or broth color or whatever other color you want to call it, but not pink red is the key. And so if it is not pink red, that means that acids were not produced and that the pH was greater than 4.4, meaning the pH did not go down for the methyl red to detect the acids. So that would be MR negative. So you want to know what the positive looks like and why. What does the negative look like and why? For your VP test, you don't need to, again, memorize all of these details. I'm not concerned with that. What I want you to take home from the VP test is that we will be detecting the presence of the acidoin. If acidoin is present, the tube will turn pink red. There are two reagents, VPA and VPB. And again, those will change color with the acidoin. And so VP positive would have pink red. And what that tells us is that acidoin was detected. Acidoin is present. And so the VP caused the tube to turn red in color. If the tube is not red, so if it's a coppery color or what looks like a very, very, very faint pink, is considered a negative. That means that acidoin was not detected. And that slight color change that you see at the top is simply due to the reagents reacting with one another and not the product of acidoin. The acidoin will cause it to turn like a bright pink red on the top of the tube. So you want to know, what does a VP positive look like? What does a VP negative look like, and why? So again, you don't need to memorize which organism does which. However, if I told you these tubes were MR, methyl red tubes, you should know that this one, if this was just labeled A and this was labeled B, you should know that A is MR positive and B is MR negative. For VP, again, you're looking for that pink red. So this would be your VP positive. This would be your VP negative. You can kind of see like this pale pink or kind of copperish color there. That is not positive. That is the reagents reacting with one another. The positive is going to be this pink red on the top. And so again, for your MVIC, the first three letters, the indole, the methyl red, and the Voges Proskauer, for all three of the first test, the pink red is going to be the positive. Now, for your citrate utilization test, you want to know the purpose. The purpose is to determine whether bacteria can utilize citrate as a sole carbon source. You want to understand that the citrate agar, the slant, is a defined media. You want to know that it's a defined media, meaning that all of the ingredients that are in the media are defined. There are no ingredients that vary from batch to batch. There are no peptones, there are no yeast extract, beef extract, etc. This is defined media. You want to know, why do we add citrate to the media? Well, the citrate is our sole source of carbon. It's our substrate. And so we want to know that the citrate is there because organisms need carbon. They need to take in carbon to make all macromolecules. And so we are trying to determine if bacteria can use the citrate as the sole carbon source. Then we have the ammonium dihydrogen phosphate. You'll notice on the next slide, I abbreviate that or I condense it. Ammonium phosphate is fine. You don't have to remember the dihydrogen part. The ammonium phosphate is there because it is the source of nitrogen. And so if the bacteria can utilize the citrate as the carbon source, they will also need a source of nitrogen. And so the bacteria would then break down the ammonium phosphate and it would produce basic products, which is our readout. So again, this test is an indirect test. We are not detecting products of citrate metabolism directly, but we are detecting the products of another process that only happens if citrate is also metabolized. So again, you don't need to know that the dipotassium phosphate is the buffer. You don't need to know that we have the sodium chloride or the agar. Those ingredients are less important for you to know. You do want to know that we have bromothymol blue in there. That is the pH indicator, and again, what colors go with which pH. For this test, you again want to study your biochemical sheet. And so when you fill out your biochemical sheet for this, we have two substrates that you should record. The citrate, this is your source of carbon, and the ammonium phosphate, which is the source of nitrogen. If bacteria produce these two enzymes, the citrate permease and the citrate lyase, both enzymes are endoenzymes, they're within the cell. If bacteria possess these enzymes, then they can use citrate as their sole carbon source and they will grow. If they grow, right, they're using a carbon source. If they grow, they also need to utilize a nitrogen source. And so simultaneously, they will utilize the ammonium phosphate. You don't need to know an enzyme for this part of the process, but you should know that the ammonium phosphate would be utilized by the bacteria and it will produce the products that are detected. The products of this test are the ammonia, the NH3, and the ammonium hydroxide, the NH4OH. These are our products. And again, these are indirect products of the citrate metabolism. We're not detecting the products of citrate metabolism directly. We are using an indirect method because if bacteria utilize the citrate, they will also utilize the ammonium phosphate because they need a source of nitrogen. You should know that they need nitrogen to make proteins and to make nucleic acids. So this is why bacteria must take in a source of nitrogen. They need it to make those macromolecules. And so if they utilize the ammonium phosphate, they will produce the products, which ultimately gives us our readout. And in this case, the ammonium hydroxide and the ammonia both will raise the pH because, again, they are basic products, so they will raise the pH, and that causes the bromothiamyl blue to turn blue. You should know that what the positive looks like and what the negative looks like. The positive test is going to be blue because the only way that you get alkaline products is if bacteria utilize the citrate as the sole carbon source. Or another positive result would be green with growth. Because if we see growth, the only way that the bacteria grew is if they utilize the citrate. They cannot grow if they can't utilize citrate. So if we see growth, that is still considered a positive. Maybe the bacteria has not had enough time to produce enough alkaline products for it to turn blue. But if it grows, that is still considered a positive. That is also why we have to inoculate with a needle. We have to inoculate with a needle so that we don't pick up and transfer too much bacteria. Because if I transfer a lot of bacteria, it might appear as though bacteria grew on the slant, but it didn't. You just transferred a lot. So in this test, if you see growth, that is a positive. Your negative test is going to be your green with no growth. That is your negative. Because if they don't grow, that means that bacteria did not utilize the citrate as the sole carbon source. And so that's what makes this test different than, say, a Fennel Red broth test. Because in this test, no growth is the true negative. That's the negative in this because the only source of carbon is the citrate. If bacteria don't utilize the citrate, they also don't grow. In the case of the Fennel Red broth, no growth was inconclusive because in the Fennel Red broth, there were peptones, there were sugars, there were multiple food sources. Bacteria should have been able to use something. So in a Fennel Red broth test, no growth would be inconclusive, but in a citrate test, no growth is a true negative. Because again, if they can't utilize the citrate, they can't grow. Now again, in order for that to be meaningful, you also have to check that the cap was on loose, that you didn't screw the cap on too tight. Because if the cap was on too tight, right, then if air doesn't get in, the bacteria won't grow, and you would record that as a negative, but it's actually not. You just didn't do the experiment correctly. So this is what you need to study for the citrate test. This one is a little more complicated because it's not a direct test. Again, there's more than one substrate in this test. There is more than one product. There is more than one enzyme, but you want to study all of those. And you want to know, what does a positive look like and why? And what does a negative look like and why? So this, again, is just showing you the different results. You don't have to memorize which one E. coli was and which one Enterobacter aerogenes is, but you should, if given tubes, be able to tell me whether or not that is a positive or a negative. And so again, blue with growth would definitely be a positive. Green with no growth would be a negative. And so you just want to review these and practice and make sure that you could look at these tubes and be able to tell me which tube is positive and which tube is negative. And so this will conclude our review of Quiz 2.